Hi, I'm Sam, a co-creator of CoMarker, an end-to-end image analysis pipeline produced as a part of the UK Dementia Research Institute Multiomics Atlas project. The aim of this tutorial is to show how accessible CoMarker is and how easy it is to use. A full instruction manual can be found through the link on our GitHub repository. To download the repository, click Code and then Download Zip on GitHub. Once this is finished downloading, drag the download onto your desktop or wherever you want to save it. I have decided to just rename the repository to CoMarker, just so that it's better presented on my desktop. Once this is done, we can close the GitHub tab since this is no longer needed and take a look in the folder. Here we can find all of the functions used in CoMarker. We can find the regular functions and the bash script. We can also find a sample directory with images and metadata that we have provided. You can practice using CoMarker with these and kind of get to grips with how it works. The image folder is organized such that each folder within the directory is a technical replica of one patient. Each replica of each patient, of each case ID, will have its own folder, and in each folder you'll have images labeled by whichever marker they are. We have also provided a sample metadata folder. At a bare minimum, the metadata file needs a case ID column as well as an outcome column. The metadata file must be saved as a CSV. Now let's start running the pipeline. So first of all, we need to open Fiji image.j. This is where the first part of the analysis works, and we'll open the CoMarker macro file. Now we'll click Run, fill in the parameters. We're able to set the marker, minimum size, thresholding method, and various different parameters that we have. You can find more details on these in the manual. Again, next page of parameter selection. We've tried to make it so everything is customizable. This way, the pipeline can really reach its potential and be used in a variety of ways. Once all 3 pages of parameters have been selected, click OK, and this will take you to choosing your image directory. Find the image directory in the repo sample file. It's in the sample directory, and then click OK. Now the code will run. We can see in the summary table what the code is analysing. In this run of the code, we've used amyloid beta as the region of interest. We're also counting how many nuclei there are. We're analysing the IBA1 signal, as well as when that signal is within 2 microns of a nucleus. We're also analysing the reference marker signal within 2 microns of the nucleus, within range of the region of interest. In the sample run, we have used CD68, a microglial activation marker, as our general colocalisation marker. The data we get about this marker is the same as that we get about the reference marker. However, we also analyse the colocalisation of this marker with the reference marker, both with and without respect to the region of interest. It is very useful to have a read of our manual, which you can find online. This will tell you more about the exact steps that are taking place in this analysis, how we're counting cells, how we are classifying the region of interest, and the different optional functions that you can use. It's definitely worth a read. It can definitely help you make the most out of the script for your own personal use. It is important to understand and customize the parameters so that they work best for your markers. The Gaussian and watershed function we have for the region of interest works great for our amyloid beta regions of interest, but not necessarily for others. So definitely worth testing it out, test out different thresholding methods and test out minimum sizes. So now this stage of the analysis is done, we can close Fiji image.j and open R. We now need to open the wrapper function, which is found in the functions folder in the repo. And there is also a sample input parameters file, which you can use, or you can type in the input parameters yourself. So the first step is to run the wrapper function, and then we can go to the input parameters section. We need to input the directories and the other parameters. The outliers threshold parameter allows you to input the amount of standard deviations away from the mean you're going to accept as not being an outlier, and remove outliers being true, means that anything that is more than 3.5 standard deviations from the mean in this case will be removed from the analysis. Now that we've run the script, we can go and find the report, which will be in the results directory in a report file. We can open the report and it gives us lots of useful information. It tells us how many plots were produced, how many significant comparisons there were, how many samples, case IDs, technical replicates there were. Let's scroll down here and we can see the box plots with annotated statistical significance. We can use the contents menu on the left to skip to different parts of the report. This is organized by marker, and we can actually skip to the data section, which allows you to save the data as CSV, Excel, or copy it. You can change which columns are visible there, and you can search for data. You can search by replicate, case ID. There is also a section for flagged images. Nothing is appearing here because nothing was flagged. However, if you do have any poor quality images, the code will pick them up and present them here. I hope this tutorial has been useful. For any extra information, check out our manual, which you can find the link to on GitHub. The manual contains step-by-step instructions for running the pipeline, either using Fiji and R, as I have just shown, or through the terminal. Thank you very much for watching this tutorial. We hope that you find CoMarker easy to use and that it can make a difference in your projects. If you do have any issues, raise them on GitHub and we'll strive to get back to you as quickly as we can.